So we're going to start the macromolecules with carbohydrates or carbs. So carbohydrates can range from really small sugar molecules, which are just monomers, to large polymers, which are polysaccharides. So the single monomers are called monosaccharides, and the large polymers are called polysaccharides. So those are the two words that are in your chart that I had flipped past there. And so those are kind of the key words. Sometimes with these, coming up with examples are really, really useful for kind of cementing your knowledge. So basically what we're talking about with all these monosaccharides are the main fuels for cellular work. So glucose is a good example of a monomer or a monosaccharide when we talk about carbohydrates. If we were to talk about the concept of a compound and that fixed ratio business, all carbohydrates, doesn't matter what it is we're talking about, fit into this 1 part to 2 parts to 1 part, though that fixed ratio. So glucose, if that was my example, is actually C6H12O6. You could still reduce this to the same fixed ratio. So like if you make rice and it's a 2-to-1 ratio, you can make more than 1 cup of rice, but you can make 6 cups, but then you'd have 12 cups of water. That's the idea here. If you were to take a look at glucose and its structural formula, I mean, it's a complicated molecule. In our work, it gets kind of in all the schematics and stuff, it gets abbreviated to this sort of purpley mauve type shape there. And so that's what our textbook uses every time that they're talking about glucose. So we have the idea of a monosaccharide and a polysaccharide. In between, if you have 2 monosaccharides or a coming together, then that's called a disaccharide. A good example is a glucose and a fructose monomer coming together, and that's table sugar. I was going to try and pull one past you here. This was the example from the text materials where it's a glucose and a glucose not coming together to make fructose, but another carbohydrate, another disaccharide, which is maltose. In these ones, you can see something that we have seen before, where you have those getting out of the way. So it's a dehydration synthesis reaction, right? Where the water comes out, a condensation reaction because the water's coming out. And those come off and the water comes out so that the two molecules can actually bond together. And so that is an element like we were looking at before with the beads becoming a longer chain. And so the same process is going on. So polysaccharides are polymers of monosaccharides. It can be more than 2, it could be 3 or hundreds. And they basically have 2 different functions, these polysaccharides. Some of them are used for energy storage and some are used for structure in a compound. So there are some important polysaccharides in biology, and basically these next 2 slides talk about compare and contrast. So starting out, how they're all the same, all of the molecules, all the important polysaccharides that we're going to talk about are all polysaccharides, are all polymers of the monomer glucose. So changing my color to that sort of like mauvey type color, I think that's the way it was drawn, right? They're all these glucose monomers linked together, right? And so those are the repeating beads in the polymer. Taking a look at how they're different, we could talk about those 2 key jobs, so energy storage and structure. As we go down the list here, they're grouped into energy storage for plants and structure for plants, and then energy storage for animals and structure for animals. So kind of noticing that pattern can help when you're trying to get your head around all of these. Now I'm going to go back and take a look at the words. So the words here that we're talking about, the different kinds of polysaccharides would be starch, and I think of potatoes when I think of a starchy something. And so the potato put that polymer of glucose molecules in itself to store energy for the potato plant. Now, I eat them and get energy out of them, but they really are for energy storage in a plant. The parallel to that is in my liver is glycogen. So I know that I have a bad habit of not eating breakfast. And so when I get up and I have a double espresso and then don't eat anything till about 10 o'clock, my body is working off of glycogen. That's a polysaccharide of glucose that my body stored in my liver after I ate dinner the night before. And so we hydrolyze that, we break up the glucose when we need it. So when I'm not eating breakfast in the morning. So these are both energy storage, one in plants and one in animals. Again, paralleling that, if we talk about the structural polysaccharides, cellulose in plants gives, a woody structure. It's why stems stand up. A plant has integrity as a plant. And then this is in animals. So chitin is in animals, but not in humans. And so if you were to think of an exoskeleton of a crustacean or an insect, a beetle or a lobster, the material that their exoskeleton, their armor is built out of is a polysaccharide called chitin. So we've now talked about how the polysaccharides are the same and how they're different in what they do, right? Their functional difference. Let's take a look at the next picture one because these are structural differences. And so working through them in the same order I find useful. And if I summarize these pictures, we've got starch is that monomer of glucose. You can see, right? They're all monomers of glucose, right? Starch is a coiled structure where glycogen is both coiled and branched. I don't know if that looks like it to you. Cellulose and chitin are very similar. They're harder to digest, which is why the cellulose is fiber in our diets, right? Because we don't digest that. And the reason for that is that they're these straight chain polymers, and they're actually fortified by a secondary bond. And so that makes them harder to break down, and that's why when you eat that structural polysaccharide, you don't break it down. It comes through you as fiber. So always with compare and contrast, you want to say how things are the same and how things are different. And these two slides say how they're the same and how they're different, both in function and structure, in function and form, if you will. Remember, form and function is one of those themes. All right, let's go to the second one. So protein is kind of your default answer in this. Proteins are everywhere in biology. And maybe I should switch to a different color here because I had my glucose purple on. A protein is a polymer built from combinations of 20 different amino acid monomers. So monomers are amino acid, and you'll find that in the chart. And the polymer gets a special name based on how the amino acids bond. So the polymer is a polypeptide. I'm going to kind of plant that here, but it's also on the next slide. It's involved in nearly every dynamic function. That's why it's your default answer. And they're super diverse, and shape is everything when it comes to proteins. There are so many functions and so many different kinds of proteins. I picked some that are relatable, I think. So enzymes, and I'm thinking digestive enzymes, in addition to enzymes that make chemical reactions go in your body. But digestive enzymes are a big one. Antibodies, that your body produces, are just little proteins floating around in your blood that help with your immune system. Cell identifiers, this helps if you know your blood type. So if you have blood type like I do, I have blood type AB, that means that I have little a protein markers and little b protein markers on all my blood cells. If you're a type O, that's actually a 0. That means that you have an absence of those cell identifiers on your blood cells. So many cells have identifiers, and blood type is just one example that you may know or may have heard about and can connect to a little bit. Cell transport, so that selectively permeable membrane, how things go in and out of a cell. Proteins are kind of the gateway on that. Hemoglobin is a transport protein in your red blood cells, and its key job is helping transport dissolved gases in your body. And then muscle contraction, we're talking about proteins when we talk about how your muscles actually contract and shorten. So great examples of proteins. If we were to talk about proteins and that lens of monomer polymer thing again, the amino acid is the monomer. And basically, an amino acid has two ends. It has an amino end, so we're talking about functional groups here, so the amino functional group and a carboxyl functional group. And sometimes carboxyl functional groups are called carboxylic acid. And so I put them in red. Yours don't copy in red. This is how we get amino acid as a name. They're named for the two functional groups. Oops, it's right there, of the organic molecule. So amino acid monomers are linked together to form polymers called proteins. And they're typically, the polypeptide is what we talk about with the protein. So a polypeptide. It's a word that I kind of think isn't in the notes enough early enough on, so I'm adding it as we go through. So this is kind of funny too. It's a whole chicken and egg situation. In order to make a protein, you need a protein. And it's a protein-mediated dehydration reaction. If you think back to what that means, it means that water is coming out so that the two amino acids can actually link together. So that dehydration reaction links the carboxyl group of one and the amino acid group of the next amino acid to make a dipeptide, or two amino acids linked together. They're a dipeptide because the peptide bond is what's holding the amino acids together. So I like pictures. Hopefully maybe you switch to that next slide and the picture. Here we have two different amino acids, and they're different only in this R group here. So we'd have 20 different R groups, or 20 different amino acids, but they're the same because they all have an amino group, right? And they all have a carboxyl group, or a carboxylic acid group. And so these neighboring functional groups come together. We have an OH and an H coming out as HOH, or H2O, so that's a dehydration reaction. And that link right there that's holding the two together is called a peptide bond. So this two together would be a dipeptide, many together would be a polypeptide. And you can use those two words somewhat interchangeably. The better word for the polymer is a polypeptide, but when we talk about a polypeptide, you could replace that with protein if you like. Okay, let's keep moving on. Shape, like I had said, is everything with proteins. And so the shape of the protein determines its function. And again, this is a theme from back in chapter 1 that we were talking about. If for some reason a protein shape is altered, it will no longer function. That process is called denaturation, or it denatures the protein. Basically, it makes it unravel or lose their shape and therefore their function. Now, you think of that probably as a bad thing, but when I cook my food or bake or preserve food like pickles or something, I'm using that concept of denaturing proteins all the time. So you can manipulate proteins by changing the environment that they're in. You can change the pH, making it really acidic, like vinegar and pickles, really salty, again like pickles. And what this is doing, it's not denaturing the protein in the pickles, or sorry, in the cucumbers. It's denaturing the proteins in the microbes that might cause the pickles to rot. And so this is why it preserves. Oftentimes it's heat that we're talking about when we're cooking things. And that definitely happens when you cook an egg. You're definitely denaturing a number of proteins. Yeah, it's really cool in food chemistry when you think about denaturation, or denaturing of protein. Our third macromolecule would be nucleic acids. Nucleic acids, the classic DNA, NA is standing for nucleic acid. DNA are your genes. They're inherited from your parents, and they basically, this is the cool thing, they provide directions for their own replicating. So their own replication, they're self-replicating, which is really awesome. This is a unique feature of nucleic acids. Polymers of carbohydrates don't self-replicate. Polymers of proteins don't self-replicate. Molecules of lipids don't self-replicate. So this is something that's unique to nucleic acids. And really, it's the programming that's in your cells. They direct the synthesis of proteins. So we have DNA works through an intermediary RNA to actually make proteins. And that's how it works in your cells. So these are both nucleic acids, and they have different letters in front because they have a slight difference in their chemical structure. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. The way that they're different is that they have a different sugar in them. So this has ribose as a sugar, where this has ribose minus an oxygen, so deoxyribose. Almost the same, just the element of oxygen different. And so these both have monomers called nucleotides. So we could talk about, again, that chart. The monomer of nucleic acids are nucleotides, and they're going to come together. The polymers are called polynucleotides. So all nucleotides have three parts. We have the phosphate functional group. We have the sugar, either ribose or deoxyribose. And then we have up to four different nitrogenous bases, four different kinds. This is where we're talking about all that diversity in genetic makeup and a very limited set of monomers. DNA has the nitrogenous bases adenine, thymine, cytosine, and guanine. You do not need to memorize a lot of this stuff. I like summarizing them just into the letters. And the idea here is that the monomers link from their sugar and their phosphates. And so what we end up with is kind of like the rails of a ladder, this sugar phosphate backbone going up the side with protruding nitrogenous bases. So here we have the cytosine, the thymine, the adenine, the guanine, right? And so you can kind of like this box is around a single nucleotide. The nucleotide had three parts, the phosphate, the sugar, and that protruding nitrogenous base. And so as we link the sugars and phosphate groups, we end up with a repeating sugar phosphate backbone with protruding, like rungs of a ladder, nitrogenous bases. So it does one more thing. DNA is not just a polynucleotide, but it's actually a double polynucleotide. And it's also a twisty one. So it's a double polynucleotide makes that DNA double helix. And this is the iconic image. I mean, you don't need to study biology in order to see this image out in the world, but this is what we're talking about. So this would be a polymer of DNA. There is specific base pairings. If you play Scrabble, the only word you could make out of A, T, C, and G, unless you're playing with the Scrabble dictionary, would be at. Just two-letter words, right? That's all we can do. And so A's and T's pair together and C's and G's, maybe because they kind of look alike, just as letters, also pair together. RNA is a little different, so it also has, like I said, a different sugar. It's also a single polynucleotide, so it doesn't have this double helix structure, and it has one different nitrogenous base. It has a U instead of a T. And so, they're definitely linked. They work together. There are definitely similarities, but there are some differences between DNA and RNA. If we were to, again, summarize how they work, a particular nucleotide sequence, remember that's the monomer, can instruct the formation of a polypeptide or a protein. So that sequence is called a gene. And you might have genes that code for eye color or hair color or any of these things, right? Um, Mostly we have millions of base pairs and many genes in DNA molecules. Um, they're really what gives all of the variability to us. And this is the overall flow. DNA uses RNA to make polypeptides. And again, with that, if you don't like the word polypeptides, you can always write in protein. This is where your genetics are actually expressed. And so this is coded in you, and this is what you can see, right? That's the expression. It's how it's translated literally into who you are. So the last one, lipids. And again, with lipids, you might think dietary fats, but it goes beyond it. Lipids are the only things that are not a polymer, and so that's noted on the chart. I included it in the chart and put that. There are fats. There are phospholipids, which might be something new to you, but are very important. And then in the notes, it also talks about steroids. So lipids are mostly insoluble in water. They lack that real like hydrophilic functional group that makes them soluble in water. So fats, as we think of, you might think of a triglyceride. A fat is a large lipid molecule made up of two smaller molecules. So we have a glycerol and then a fatty acid chain. If it's a triglyceride, there's three fatty acid chains in a triglyceride. Well, that's really squishy in there. The thing about fats is they contain twice as much energy per gram as does protein or carbohydrate. So their real main job is energy storage in our bodies. Take a look. I put a little note here. It's a dehydration synthesis that's linking the fatty acid tail onto that glycerol molecule. So in as much we're not talking about monomers linking to polymers, it's still a common chemical process in these macromolecules that we're learning about. So you can have two different kinds of fatty acids. So this would be a triglyceride. And remember how we talked about how carbon can be single bonded or double bonded? So if it's single bonded, then it's got the maximum number of hydrogens filling it up. If it's unsaturated, it's not fully full of hydrogens. And so here you could break the double bond to put more hydrogen in. So unsaturated has double bonds. Saturated, no double bonds, only single bonds. And so basically we think of oils as these unsaturated fats. Those double bonds prevent them from packing tight into a solid like butter, lard, coconut oil, these kinds of things. Where saturated fats, the fatty acid tails would go straight and allows them to come together. So hydrogenated vegetable oils are what were a way of making or are a way of making margarine. And so basically, I don't know how to spell margarine. AR? I don't eat margarine much. So unsaturated fats converted to saturated fats by adding hydrogen. They're hydrogenated vegetable oils. And so you can make them solid as a butter replacement when chemically they are, not what butter is. So phospholipids are really important. These are the ones that I said are kind of new. They're all about the membranes. We're going to see this as we keep through to the chapter 4 and chapter 5. This is a phospholipid bilayer. Each of these molecules with a head and two tails hanging down is a phospholipid. The cool thing about a phospholipid, it's kind of like the fat molecules too. These heads are water loving. So the fancy word is hydrophilic, right? The tails are hydrophobic. They're kind of like those fatty acid tails where they're just hydrogens and carbons. They don't dissolve in water. So this is water hating. So what happens with phospholipids is something really cool. They will auto-arrange in an aqueous environment, in a water environment, to make this phospholipid bilayer. And so this is the protective layer that makes every membrane in a cell. So all cell membranes are phospholipid bilayers. So you would just be a pile of goo if you didn't have these molecules giving your cells integrity or giving the organelles in your cells integrity. And so this phospholipid bilayer will not go away anytime soon. But it's important as we get introduced to lipids. So we've done lipids, which are fats, phospholipids, and then the last one here, which are steroids. And this is a good example of, understanding some chemistry. Steroids are lipids composed of fused ring structures. So going back to that organic molecules that can make rings. Cholesterol is an example of a steroid that plays a significant role in the structure of cell membranes. It basically, cholesterol is often given a bad rap, but on a cellular level, we need cholesterol to basically keep our cell membranes fluid. In addition to cholesterol is an important compound from which we synthesize some sex hormones. So not all sex hormones are lipid-based, but some of them are. I'm going to leave you with this. This can be helpful. It is in the review. I'm happy to take it up, and it's also a really useful tool as we work through the conclusion of Chapter 3 and the first activity in the lab.